Before the antibiotic penicillin was discovered, staph infections commonly led to death. Our grandparents survived these infections because of penicillin. But did you know that if you were to contract that same infection today, that penicillin would no longer be an effective treatment option? This is because the bacteria that causes staph infections are almost completely resistant to it, meaning the drug no longer kills them. Antibiotic-resistant bacteria pose a serious threat to humanity, and the problem is only growing. Despite this, development of new antibiotics is actually dwindling. A major reason is that it's just really difficult. A good antibiotic must meet a couple of key criteria. One, they must be able to get into bacterial cells. And this is not trivial. Bacteria are amazing at keeping things out of them. Two, they must kill the bacteria by targeting something specific to them, but not harming human cells. Other researchers working on developing new antibiotics may use methods that are too broad or random to efficiently identify good drug candidates. I have developed a streamlined approach to screen thousands of chemicals and specifically hone in on antibiotic candidates that meet the key criteria. I exploit these natural sensors called riboswitches that some bacteria use to sense changes in their environment. They bind these specific signals of stress represented by stars in the diagram. When the signal binds, they act as a switch that tell the cell to respond to the change. These are very similar to the warning lights in our cars. Just as our cars may sense and alert us to different issues such as an engine problem or low oil, different riboswitches sense and alert bacteria to different types of stress. I've introduced my own readout to this system that causes the bacteria to turn blue when the stress is detected. I can then use a riboswitch as a tool to screen thousands of chemicals and specifically identify the ones that are able to get into the bacteria and stress them in a bacteria-specific way, which I can then detect using the color change. The chemicals that I identify can then be developed into new antibiotics. My grandchildren might not be able to use the same drugs that I would be prescribed to treat an infection, but this method will help ensure that they have new drugs to help them in our arms race against bacteria.